Hello to our friends joining us via recording. It is our last review session before exam number 2. Remember, if you were not able to attend this session or the session that we had before spring break, there are recordings for you to watch of that review session as well. To start off today's review, we worked in groups to work through the process of the Central Dogma. And remember, the name, the Central Dogma, sounds really important, really big, yeah, scary, but the Central Dogma is actually not that scary. The Central Dogma is just the idea that your cell stores information as DNA, and it uses that DNA to build proteins. So for us to take our instructions in DNA and use them to build proteins, which is what translation does, we have to go through this middle step called transcription. So when I have a DNA sequence and I do transcription on it, what do I call the thing that I make? When I make a sequence here, what's that sequence called? In transcription, we're building, yeah, exactly. A couple of us have chimed in. In transcription, you build mRNA. So I was chatting with one of my groups. On the exam, I could ask you either to do the process of transcription, which is what I ask you on this assignment, or I could ask you, I'll type the whole thing out, to build complementary mRNA. Either of these are asking the same thing. If I ask you to build complementary mRNA, or if I ask you to do the process of transcription, that's the same thing. When I do transcription and I'm building my complementary mRNA, I have this word complementary that I'm using in it. Remind me, complementary means we are or are not exactly the same. The sequence are or are not exactly the same. Yeah, when we are complementary, we are not exactly the same. We, as a couple of us mentioned in the chat, are mirror images, or I can use the sequence of one genetic information, the DNA, to predict the sequence of the other set of DNA, or excuse me, of RNA. So when I'm going through and doing transcription, one of my nucleotide bases, I cannot include it in RNA. Which of these letters, A, T, C, or G, who's too cool to be in RNA? Who can't be there? Yeah, exactly. T is just too cool for RNA. Thymine, you will never find that in RNA. What letter do we find in place of thymine? Who's actually able to be in RNA? Yeah, exactly. I can use uracil in RNA. So here's another thing I chatted with the group about. What we've done in the past, especially when we were working through this activity, is we started with our DNA sequence and we built complementary DNA to go with that DNA sequence. When we build complementary DNA, we can still use thymine. If on your exam I ask you to build me DNA, complementary DNA, you still get to use T. T gets to be in there. If I ask you to build me complementary RNA, that's when we can't use T. So please check when you're taking the exam. Read your questions slowly. Make sure if I ask you for DNA, you don't give me any uracils. There's no uracils in DNA. Make sure if I ask you for RNA, you don't give me any thymines. There's no thymines in RNA. So let's go through then, and we will work a little bit together as a class, and then I'll type most of it for me. When I am looking at my letters here, my first letters are T, A, C. Remind me, when I'm going into RNA, if I have a T that's in the DNA, what letter do I put in the RNA? If there's a T in the DNA, yep, I get to put an A in my RNA. So here, let's add our A. Notice my next letter in the DNA is an A. If I have an A, what would I put in my RNA? I can't put that T, right? Yeah, so this is when I have to put a U in. I can't put in that T because thymeine's too cool for RNA. Okay, and I've got a C in my DNA. What letter would I put in the RNA if I've got a C? Yep, now we got a G. Oops, add my G. Okay, so we can go on through here. We've got a U, space a little bit, another U, a couple of A's, and then I'll pause because I've got a G in my DNA. What letter goes with G in the DNA again? We haven't done that one yet. G pairs with C, absolutely. Yep, C is fair game. That letter we don't have to change. So let me go through and I'll get us a couple more. G. I'll have you all check me once I type it. Hopefully I stay on track. Scoot over. A. Okay, let's see how Dr. Hollis did. So we've got AUG, UUA, ACU, UGU, CGUUAA, CAUACA. Awesome. You think that's good? We thumbs up? I think we get it. Awesome. Okay. So we just did transcription. Transcription where you take the DNA sequence and you turn it into complementary mRNA. If on the exam I had asked you to do the complementary DNA, the sequence that we have here would be exactly the same, except one of these letters would be gone. Which letter would I not have in complementary DNA? I know we said it before. We're going to say it again. Yeah, we're not going to have the U. So if I was going to build complementary DNA, instead, all of these places that I had U's, I would just trade those out for T's. So that's why in class, when we were working through this process together, a lot of times we first made the complementary DNA, because then we could just take that sequence and sub out those U's. But you don't have to do complementary DNA if I just ask you for RNA. You can just go straight to it, give me those U's. Now we get to our second step of the central dogma. And the second step of the central dogma is translation. The goal of translation is to make proteins. So this is when we actually are going to use genetic code table to tell us which amino acids, those building blocks of proteins, which amino acids we're putting into our chain. When we do translation, how many of these letters do we read at a time? So in translation, here's my technical name, my letter. I'm reading codons. Yeah, so codons are groups of 3 letters. Remember, when we do translation, our ribosomes who do that, they're not very good at reading yet. So they can only read things 3 letters at a time. When you're doing translation, I highly recommend that you split up these sets of letters into groups of 3, because that's going to help you to know what exactly you're looking for on your table. So I don't have my code table in front of me. Remember, on the exam, I will 100% give you one. So do not memorize the table. With your table out, I need you to help me out. Let's start with our first codon here, AUG. On your genetic code table, what codon did AUG, or what amino acid did that go with? Yeah, so that goes with the amino acid MET, and I think it's actually colored in red on your table. MET is for methionine. That's what we call the start codon. So every one of the proteins in your body always starts with MET, with methionine. The next codon that I have is UUA. Using that table, which amino acid do we put in for UUA? Okay. Got some friends telling me that's the amino acid LU. It stands for leucine. I know I got this question, I think, last time in class, if I'm going to ask you to know their whole name or just their abbreviation. The abbreviation is all you'll need for the exam. It's just like those homework questions where I'll give you the drop-down menu with all of the amino acids. So as long as you can match up the abbreviations, you're good. Okay, so first codon, methionine. Second codon, leucine. Here's my third codon. We use our table for ACU. ACU gives me threonine, THR. Okay, let's go to UGU. That's my next codon, UGU. Okay, that one's going to give me cysteine, cis. Okay, we're at CGU now. Help me out. CGU. Okay, arginine. UAA. Yeah, okay, so UAA. On your table, that says stop. Remind me, because we're just finishing spring break. I don't remember what happens at a stop codon. What does a stop codon mean? Stop is not an amino acid. In case we were wondering, we don't have any amino acids named stop. Yeah, so one of the things that happens is, Kaylin mentioned, we're done reading. We don't go any further in our sequence. And Kira kind of explained why we don't go any further. Because when a ribosome bumps into a stop codon, remember I told you it's kind of like ribosomes, they call out to those tRNA molecules. tRNA helps it to read these sequences, except none of my tRNA molecules read my stop codons. So my ribosome will call out UAA 3, 4, 5 times. Nobody answers, and the ribosome can't even. It literally falls apart. So when the ribosome falls apart, we're not reading anymore. We stop at that point. The process is done. Check this out. I still had some codons over here. Even though there's codons after my stop codon, I don't get to read these. The ribosome doesn't get this far. So this is why it can be really problematic if we have an error in our DNA or an error in our RNA. If that error leads us to put a stop codon in earlier than it's supposed to be, we ignore everything later. And it could be that there were just 2 codons, or it could be, if we're building a huge protein, there could be 1,000 codons after that we're just not reading. So when we have an issue in our RNA, if we put in a stop codon too early, we're not going to read anything after that. The opposite could be true too. Maybe your cell always wanted to stop reading the sequence right here. But if we change this, instead of having UAA, if this codon was actually this one here, we just keep going. So stop codons are really powerful. They tell a cell when it's done building a protein, but they've got to be in the right place. We don't want it in the middle of our sequence. We don't want it till we're at the place where we're actually done building the protein. So hopefully, when we were working with our groups, when you hit this stop codon, you said, sweet, we don't have to translate these, because you don't have to at that point. At that point, we're done. Your cell is going to ignore any messages after here. How do we feel about doing transcription and translation? Thumbs up, thumbs down, crazy eyes. Good. Okay, lots of thumbs up. That's excellent. I'm promising you, just like on the homework assignment, just like that review assignment where I had you go through and practice typing in the letters for transcription and practice typing in the letter or finding those amino acids for translation, you're going to have a question just like that on the exam. So make sure to use that review assignment to go through and keep practicing doing this. The more times you do it, the more sequences you're going to see. Yeah, so Elavina asked a good question. If we hit a stop codon when you're doing the process of translation, that is correct. We would there will be an option mixed in with all those amino acids that says stop. So you would just select stop, and then we don't do anything after that. We would just leave those ones blank. Yeah, good question. Kaylin says that the central dogma is what she would rather do than some of our other things. So you're gonna love that part of the review, right? The review of the exam. Transcription and translation. Okay, let's do one more thing with transcription and translation. We're going to predict what would happen in our cell if we were missing each of these things. So let's start with this molecule here, tRNA. Remember, its name technically is transfer RNA, but I told you to think about it as translator RNA, because thinking about it as translator RNA helps you to know which process it's involved in. What do I use tRNA to do? Transcription or translation? tRNA. Get my pencil. Yeah, exactly. tRNA is my translator, right? So when we talk about tRNA, at the top it has an amino acid, and at the bottom it has nucleotides. I'm spelling that right. Nucleotides. So it's a translator from the language that RNA speaks into amino acids that proteins are made out of. If I did not have tRNA, I would get a message that tells me what I'm supposed to build, but I wouldn't be able to translate it. So the process of transcription is when we build a message. tRNA isn't involved in building a message. So I could still do transcription even if my cell had no tRNA. But I can't do translation without tRNA. So if this was missing, if your cell had no tRNA, we would not be able to do the process of translation. We could still build RNA, we just couldn't read it. But hey, let's go down to this one right here. RNA polymerase. RNA polymerase. Its name literally tells us what it does. What does RNA polymerase do? RNA polymerase. Yeah, so RNA polymerase is what I use to do transcription, right? This polymerase word means that I build something, a polymer that has multiple pieces. And what it is I'm putting together are nucleotides to build RNA. RNA polymerase is the enzyme that literally builds mRNA. The name of the process where I build mRNA, what's that called? The process I use to build mRNA? Is that transcription or translation? When you're building mRNA. There we go. I was going to say, I really hope that more than just two of us know that. Yeah, we're good. It's a long word, so you're good. RNA polymerase builds RNA in the process of transcription. RNA polymerase is directly involved in transcription, right? In building mRNA. So we know that this is not going to happen. Transcription is not going to happen. If I don't build mRNA, can I turn it into a protein? Can I do translation if there's no mRNA? Yeah, exactly. I can't do translation if I didn't do transcription. So I have to, so let's make some notes for ourselves here. RNA polymerase builds mRNA. If I have no mRNA, no translation. You can't translate something if there's nothing to translate. So if I didn't have RNA polymerase, I actually couldn't do both processes. I couldn't do transcription or translation because transcription needs RNA polymerase to happen or to function. Translation needs there to be RNA to translate. So if we do anything to mess up transcription, we're going to mess up translation as well. See, that brings us to our next one over here. When I talk about DNA, is DNA involved in transcription or translation? DNA. Hey, here, we'll make it easy. 1 or 2. Transcription is 1. Translation is 2. All my friends who worked hard to type are like, that's not even fair. Transcription 1, translation 2. We use DNA. Yeah, several of us, all of us have chimed in. DNA is involved in the process of transcription. Actually, that's the goal of transcription, right? Like Kaylin's mentioning, DNA has to stay in the nucleus. I can't build proteins in the nucleus, so I do transcription to get those proteins out, or to get the direction for those proteins out of the nucleus. So DNA is directly involved in transcription. But if I don't do transcription, I'm also not doing translation. So if your DNA is missing, again, we're not going to be able to do transcription or translation. Both of my processes are not going to work. Last question talks about ribosomes. What is happening, or what is the function of ribosomes? What do ribosomes do? I talked a little bit about them, right? Yeah, so Alpine is mentioning ribosomes are involved in translation. Absolutely. Remember, ribosomes are the ones that call out to the tRNA. They ask the tRNA to help them to figure out what amino acids they should put together. So, oh, here's a good question, a tie things together question. Ribosomes attach amino acids to one another. What special kind of chemical bond do ribosomes build between amino acids? Yeah, exactly. Several of us are chiming in here. I'll put that note on our slide. Ribosomes, build peptide bonds. Peptide bonds. Remember, peptide bonds, that was one of those things I called it an underline highlight star idea with proteins. This is the special kind of bonds that we find giving us the primary structure of a protein, where we connect those amino acids to one another. So ribosomes, their job in the process of translation is to build those peptide bonds between the amino acids that tRNA brings to the ribosome. Ribosomes are involved in translation. If I didn't have ribosomes, obviously translation couldn't happen. Could transcription still happen if I didn't have ribosomes? Could I still make mRNA if I didn't have ribosomes? Yeah, I definitely could still make RNA if I didn't have ribosomes. I just couldn't read it, right? It wouldn't be very helpful to my cell, but I can make it. So if I have an issue with my ribosomes, that's going to give me problems with translation. Transcription could still happen. I'd still make those messages. I just couldn't use them. That's totally correct. Somebody had chimed in earlier, and I didn't mention it at the time, but this problem with DNA being incorrect, they tied this into osteogenesis imperfecta. So osteogenesis imperfecta, let's add some notes here. DNA, we have a mutation, right? So osteogenesis imperfecta, or sometimes you'll see it when you're working on activity, they call it OI. It's way easier to type, right? So in osteogenesis imperfecta, we have a mutation in our DNA. So when I do the process of transcription, I make an mRNA molecule that's not quite accurate to the way that it's supposed to be if I didn't have this mutation. And when I take that message that's slightly incorrect compared to normal, and I translate it, I build a protein that's really weak. It has the wrong amino acids in it and makes it really weak. What was the name of the particular protein that I make incorrectly in osteogenesis imperfecta? Yeah, exactly. Osteogenesis imperfecta is a problem with the collagen protein. The collagen protein. The goal of collagen is to make your proteins able to have a little bit of flex, or make your bones have a little bit of flexibility to them, meaning you can put a little bit of pressure or a little bit of torque and they're not just going to snap. When someone has osteogenesis imperfecta, remember that the easy person name for it is brittle bone disease. Brittle bone disease, the bones just snap. And the reason they snap is because there's not good, strong collagen to prevent them from breaking. So that's an issue with having a mutation in the DNA. Yeah, Kira mentioned the other thing that we talked about in this unit that was an issue with the DNA, and that was sickle cell anemia. Sickle cell anemia. Again, we've got an issue with the sequence of our DNA. Remember, it was just one nucleotide that was wrong, but it wasn't in that place. Remember Dr. Ollis's favorite location? The wobble baby base. It wasn't in that last one, right? So in sickle cell anemia, we changed the nucleotide that was in the middle of the codon, and that nucleotide was enough to change which amino acid we put in. What protein do we build incorrectly in sickle cell anemia? Sickle cell anemia, not a collagen problem. Yeah, sickle cell anemia is a hemoglobin problem. Hemoglobin protein problem. But the basic idea is the same, right? I do transcription wrong because my DNA was wrong. And if I do transcription wrong, I'm going to do translation wrong. So both of these processes cannot occur correctly if your DNA is not correct. And hey, that kind of ties into what we talked about with protein structure in general, right? If I've got an issue with my protein's primary structure, I might have an issue with secondary structure and tertiary structure and quaternary structure. We got to get all these processes correct for things to function. So if you mess up transcription, you're going to mess up translation too. If we mess up the primary structure of a protein, you're probably going to mess up secondary and tertiary and quaternary as well. I think someone had mentioned earlier, it's just like we talked about way back on day one. Anatomy and physiology, they depend on each other. If we change the anatomy, the physiology is going to be wrong. Or if the physiology is wrong, that might have effects on the anatomy. It's the same idea with transcription and translation. You can't do one correctly. You can't do translation correctly if we're not doing transcription correctly. Transcription, translation. Thumbs up or any questions before we move on? Okay, getting lots of thumbs up. Perfect. Kaylin doesn't want this to be on the exam, alas. Now we're prepared if we see it, right? We're in good shape. All right, let's look at another thing that we wish wasn't on the exam, but hey, if we do it now, we'll be prepared in case it is. We learned about two hormones that were involved in calcium homeostasis. Hey, homeostasis was a new idea from this unit. We talked about homeostasis in terms of body temperature, and we talked about homeostasis in terms of blood calcium in particular. So blood calcium and your body temperature, we keep at a relatively stable state. Like Kira mentioned, we keep our body at its set point. set point. So that's the normal range of values that we want to be at. When your body is working correctly, we're typically around 98.6 degrees, or we're typically around a stable level of calcium. Remember, you don't have to memorize the level of calcium. To be totally honest, I don't remember it exactly myself. So, not a number you need to know, but we maintain a generally stable calcium state. We do that using parathyroid hormone and calcitonin. Now, before we start talking about what happens if I have too much or too little, as we'll see on the other slide, I want us first to remind ourselves about some of the important information about these hormones. So let's start with calcitonin. Calcitonin. When we talk about calcitonin, remember I told you to look at this last part on calcitonin. It has in on it. Based on this in part that's in there, what might we say that calcitonin does? Does anyone remember what I mentioned with that in? Calcitonin. Where do we put calcium if we release this hormone? Calcitonin. Yeah, so I asked a bad question, but Audrey kind of knew where I was going with it. Okay, so calcitonin, this tells your body, put calcium into the bones. Calcitonin is a hormone that tells us to put calcium in to our bones. So when we put calcium into our bones, this means that bone density goes up, or bone calcium goes up. But when the calcium level in your bones goes up, that calcium didn't just come out of nowhere. It didn't just appear. That calcium came out of the bloodstream. Remember, there's kind of two places you should consider that you have calcium in your body. Place number one, in your bones. Place number two, in your blood. So if I'm putting calcium into my bones, I am taking calcium out of the bloodstream. Taking calcium out of the bloodstream. So when you're studying what calcitonin does, calcitonin tells your body we need to put more calcium into our bones. And where we get that calcium to put it into the bones is from the bloodstream. Yeah, Kaylin's totally right. We can't have it in both places. We can't have high calcium, the amount of calcium in the bloodstream going up, and the amount in the bones going up. They're opposites of each other. So if we take that calcium into our bones, it came from the bloodstream. Yeah, it's kind of like a seesaw, like Kira said. So if it's going up in the bones, it's going down in the blood. If it's going up in the blood, it's going down in the bones. So calcitonin says we need to put more calcium into the bones. Parathyroid hormone does the opposite, right? So when our body releases parathyroid hormone, what happens to the amount of calcium that's in the blood? If I spit out parathyroid hormone, what happens to my blood calcium? Yeah, so blood calcium goes up. And if blood calcium is going up, then my bone calcium, or my bone density, goes down. Now, this is the one that it's easiest to understand how it works, because when the type of cell I use to make my bone calcium go down, to make my bones spit out calcium, which cell do I use to make my bones spit out calcium? Who breaks down bones? Yeah, exactly. When we talk about parathyroid hormone activates osteoclasts. I'm going to put that last part in caps. osteoclasts. So parathyroid hormone, when an osteoclast hears this message, it says, oh, there is not enough calcium in the blood. I can fix this. I'm going to break down some bone tissue where I was storing all that extra calcium, and that's going to make the calcium in my blood go up. So in normal conditions, when your body is maintaining homeostasis, when it's staying balanced, and we spit out parathyroid hormone, what happens is my bone breakers, osteoclasts, they kick into high gear. They break down some bone tissue, and that liberates calcium or spits calcium into the bloodstream. Then I can get back to normal. Calcitonin, understanding how it works, is a little bit trickier. So I want to make sure we understand this one. When we talk about calcitonin, do we remember which kind of bone cell can actually hear the message calcitonin? Yeah, so calcitonin is kind of tricky. Calcitonin is also heard by osteoclasts. But when an osteoclast hears the message calcitonin, it deactivates the osteoclast. Parathyroid hormone told those osteoclasts, kick into high gear. You need to break down some bone tissue for me. Calcitonin tells those osteoclasts, slow down. You're working way too hard. Calcitonin deactivates osteoclasts. If my osteoclasts aren't doing their job, remember it's like that lazy coworker, right? If your coworker's not doing their job anymore, it makes you look like you are the star employee. I mean, you are the star employee, right? But then you really look like you are when your other coworker doesn't do their job. So my osteoclasts aren't working anymore because calcitonin told them to stop. That makes it look like osteoblasts work harder. They don't. They're still working normal, but it looks like they're working harder. Here's our big underline highlight star idea with calcitonin. Calcitonin still talks to osteoclasts. It's still a hormone, even though it makes it look like my osteoblasts are working harder. It's still a message that only osteoclasts hear. I think someone had mentioned this when we were talking about this before. Calcitonin is the red light. It tells osteoblasts time to stop, slow down. Parathyroid hormone is the green light. It tells osteoclasts to work harder or work faster. Both of these messages, though, are only heard by osteoclasts. Please don't tell me that this one talks to osteoblasts, because it doesn't. They're both messages that are only heard by osteoclasts. Yeah, go for it, Kira. Question? Yeah, so Kira's asking a question that relates to your skeletal system disorders packet. Yeah, so there are hormones that do talk to osteoblasts. Absolutely. One of the big ones is estrogen. That's why when you were studying about osteoporosis, remember how they said that women who go through menopause are at a much higher risk for osteoporosis, because estrogen is a message that talks to the osteoblasts and tells the osteoblasts to get to work. So estrogen in females, testosterone in males and females, but primarily in males. And then, yeah, the other one Kira mentioned is growth hormone. Growth hormone, obviously it helps you to grow, right? So it's going to tell your osteoblasts to build some bone. So yeah, here we'll add a little box on our page here that says osteoblast messages. That's going to be estrogen, testosterone, and growth hormone. Those are messages that the osteoblasts can hear. But the osteoblasts can't hear parathyroid hormone, and they can't hear calcitonin. Kind of like how your dog can hear stuff that you can't, right? These are messages that your dog can hear that you can't hear. Parathyroid hormone and calcitonin. Any other questions before we start talking about too much and too little? You tracking with me? So Kaylin, we kind of need to know all of what we've talked about. She asked what we need to know for the exam about this. So we need to know what these hormones do to blood calcium, bone calcium, and the cells that they talk to. And then, about these ones up here, what we need to know about them. Yeah, so know that these ones talk to osteoclasts. Know that these ones talk to osteoblasts. And when they talk to osteoblasts, they tell them to work harder. So estrogen, testosterone, and growth hormone, all of these are green lights. These are building messages for osteoblasts. This is what we would use to make our osteoblasts work harder. These other two that we talked about, we just need to know what they do to osteoclasts. All right, so we defined for ourselves normal situation. What is the goal of these hormones? Now let's do what Dr. Allis always makes you do, right? And we're going to do some applications. We're going to talk about what happens if I have too much of these messages. So let's start with parathyroid hormone. We figured out together that the job of parathyroid hormone is to activate osteoclasts. Activate osteoclasts. If I activate my osteoclasts too much, what is going to happen to the amount of calcium in my bones? If my osteoclasts get a little too excited, if they're pulling my 4-year-old, yeah, if my osteoclasts get too active, oh, my typing's not working here. Okay, can we hear me again? I think that my computer died on me. We lost our text. So sorry. Okay, so we're just gonna have to remember. Okay, so if my parathyroid hormone, if I have too much of it, I'm gonna have well, here, we'll start this. Overactive osteoclasts. That's the first thing that's gonna happen. Because remember, parathyroid hormone is a green light for my osteoclasts. So my osteoclasts are gonna go nuts. And like you all were telling me in the chat, if my osteoclasts go nuts, they're going to break down bone tissue. And if we break down bone tissue, I'm going to have low bone calcium. Low bone calcium. Okay? But that calcium is not just disappearing. It's not going nowhere. When the calcium leaves my bones, where does the calcium go once it leaves my bones? Yeah, so if calcium leaves your bones, it's going into your bloodstream. So I'm going to not have enough calcium in my bones. My bones are going to be weaker, low bone density, but I'm going to also have high blood calcium. My calcium is not just going to disappear. It's got to go somewhere. It's going to go into my bloodstream. So the name of the condition where you have too much blood calcium, check this out. This ties into unit number 1. Hypercalcemia. Hypercalcemia. Too much calcium in my blood. So if I make too much parathyroid hormone, If I'm telling my osteoclasts to go nuts and have a party, they're going to steal my bone calcium and they're going to put it all into the bloodstream. And then I'm going to have too much calcium in the bloodstream, not enough in my bones. That's what happens if I make too much parathyroid hormone. When we talked about calcitonin, my text went away, right? So remind me, what message does calcitonin send to osteoclasts? Is calcitonin a green light or a red light for osteoclasts? Yeah, calcitonin is my red light, right? So if I make too much calcitonin, I'm going to have underactive osteoclasts, meaning my osteoclasts are not going to be breaking down any bone tissue. We only talked a little bit about this, but one of the things that we use osteoclasts to do is the process called remodeling. Remodeling, that's where you break down old bone or weak bone or bad bone, and we use it to build new bone. If I spit out too much calcitonin, I'm actually not going to do any remodeling, right? Because the cells that help me to break down bad tissue to build new bone tissue, I'm not breaking down any of the bad stuff. I just got the bad stuff staying there. So my osteoclasts, they're not going to do their job anymore, which means we're not breaking down bone tissue. So let's put a note here, not breaking down bone tissue. If I don't break down bone tissue and I'm still building it at the normal rate, because remember, my osteoblasts are oblivious. They just keep trucking along. If I don't break down any bone tissue and I keep building bone tissue, what effect, let's start, is that going to have on my bloodstream? If I'm not breaking any bone and I'm building my bone, is calcium going to go up or go down? I'm not breaking any. I keep building it. Yeah, overall, that's going to lead to low blood calcium, because breaking down bone tissue is how I get calcium into the bloodstream. So if I don't break any down, but I keep putting it into my bones, the amount that's in my bloodstream is going to go down, because it's all being put, we'll put high bone calcium. It's all being put into my bone tissue. So if I have too much calcitonin and I tell my osteoclast to just stop working, stop breaking down that bone tissue, if I just keep trucking along and building bones, but I don't break any down, I'm eventually going to run out of calcium in my bloodstream. And that's bad news for me. I know somebody put in the chat, and they're totally right, we'd call that hypocalcemia. Hypocalcemia. When my blood calcium gets too low, there's that hypo at the beginning, right? That meant low. Hyper means high. So if I have too much calcitonin, ultimately what's going to happen is my blood calcium is going to get too low. I'm not going to have enough calcium in my bloodstream. I'm going to keep putting calcium into my bones. My bones are going to have more calcium in them. Granted, I may not be building the best bone because I didn't break down bad stuff, but I'll just keep putting more and more calcium into it because those osteoblasts are just going to truck along. So too much calcitonin would mean that I wouldn't have enough in my bloodstream. Too much parathyroid hormone means I'm going to have too much in my bloodstream. And then vice versa with my bones, right? Low bone calcium versus high bone calcium. Barbara, you had a question. What was your question, Barbara? Or maybe you accidentally raised your hand?
I'm sorry. I'm having serious computer issues. Okay, so if the body is not breaking down bone, would that cause too many bones in the body because there's none being broken down?
It's not that it would lead to there being too many bones in the body per se, as much as the bones might have a higher density of not strong bones. So we build more bone tissue, and to be fully honest, we would never get to the point too where we're spitting out so much calcitonin that we break down no bone tissue for a long time.
Mm-hmm.
Your body is better than that. It corrects itself. But theoretically, it would be possible that we could build bones that are really, really huge, I guess. But again, your body.
Yeah, that's what I'm thinking. Like your bones would be excessively huge if none were being broken down.
Yeah. So if we're not doing the remodeling process correctly, we could have some issues with that, where we build bones that are getting too wide on the outside. They're not getting reinforced enough on the inside.
Okay.
So it would be bad news for us.
Okay. All right.
Yeah.
Thank you.
Caitlin's mentioning that we might look like a dinosaur, right? I actually saw I believe it's called an ankylosaurus, those ones that have like the bony back. So we could imagine that if we have too much calcitonin, we could grow our own little bony back, like the ankylosaurus. Yeah, I like it. So big picture before we switch to having too little of these hormones. If I have too much parathyroid hormone, remember that the normal job of parathyroid hormone is to tell osteoclasts to break down bone tissue. I use that breaking down to do remodeling. I use that to keep my blood calcium levels correct. If I tell those osteoclasts using parathyroid hormone to work too hard, they're going to break down too much bone, and all of that bone that they're breaking down, we're going to split that calcium in the bloodstream. So when you're making these predictions, too much of something, think about its normal job, and then just take that to the extreme. What if I did it way too much? Same deal down here with calcitonin. Calcitonin is my red light. If my light is always red, my osteoclasts are never going to break down bone tissue. So if I can't break down bone tissue, that was my source of putting calcium into the bloodstream. My osteoblasts are going to keep plugging away. They're going to keep building bone, but there's not going to be enough blood or not enough calcium in the blood. Here's what happens if I have too much of these hormones. Let's flip this question on its head now. Let's talk about what happens if I have too little parathyroid hormone or too little calcitonin. Let's start up here with this one. I'm forgetful. We just flipped the slide, so I can't remember anymore. Parathyroid hormone. Remind me again, red light or green light for osteoclasts? Parathyroid hormone. Let's make a note for ourselves. Normal level. Parathyroid hormone, yep, this is my green light. Green light for osteoclasts. Okay, which means then that calcitonin was my red light, right? So if I have a normal level of calcitonin, let me add this, that's my red light for osteoclasts. Keeps them in check, right? So if let's say, we'll start with this one because it's maybe easier to visualize. When you're driving down the road, red lights make you stop, right? Or they're supposed to. I'm teaching my daughter about yellow. She keeps asking about yellow. What does yellow mean? It's like, that's complicated. Because technically yellow means you're supposed to slow down, but if we're honest, yellow usually means we go faster. So yeah, exactly. Like multiple of you are saying, yellow means go faster, quick, get through. So let's pretend that we only have two options. Let's pretend there's no yellow. There's just red and green. Red lights, we stop at. Well, if I'm driving down the road and there's not any red lights, the red lights are all gone, am I going to go faster or slower if there's too few red lights? If I don't have something to stop at, right? Yeah, I'm just going to go really fast, right? Calcitonin was my red light. Calcitonin keeps those osteoclasts in check. So if I don't have enough calcitonin, are my osteoclasts going to work faster or slower? If I don't have enough of my red light, do these guys go faster or slower? Oop, here we're saying we have too little. So if I don't have red lights, because that's what happens when I have too little of it, I don't have my red lights, my osteoblasts are going to go faster. Yeah, I like the way Kira mentioned. Kira's saying, think about it, if I have too little calcitonin, think about it like my brakes aren't working. So I've got a car, an osteoclast car that's driving down the road, and we take out its brakes, the calcitonin. That car is just going to keep going. It's going to roll through whatever is at it. If I don't have enough calcitonin, I'm going to have overactive osteoclasts. So if I don't have functioning brakes, I like that analogy. If I don't have my brakes or my red lights for my osteoclasts, they're going to go nuts. And when osteoclasts go nuts, my bone calcium goes down because they're going nuts. They're breaking stuff up, right? And my blood calcium goes up. If I don't have enough of calcitonin, which was my brakes or was my red light that said I need to stop, my osteoclasts are going to go nuts. They're going to break down bone tissue and send it all into the blood. Yeah, I like it. Elapina made a great comparison here, and this is on purpose. If I don't have enough calcitonin, it's the same thing as if I have too much parathyroid hormone. Love it. Same idea as if I have too much parathyroid hormone. It's kind of two ways of doing the same thing. So if we think about calcitonin, we'll call this the brakes for my osteoclast, calcitonin. And we think about parathyroid hormone like the gas pedal for my osteoclasts. There are two ways to make you go faster, right? First way to make you go faster is if we get rid of your brakes. Then you can't slow down if you want to, right? The second way to make you go faster is to press on that gas pedal. So if I don't have my brakes, I'm going to work faster. If I'm pressing on the gas pedal, I'm also going to work faster. So the same kinds of things that happen when you don't have enough calcitonin are the same kinds of things that happen if you have too much parathyroid hormone. The opposite is true too, right? So if I don't press on my gas pedal enough, am I going to work faster or slower? If my osteoclasts, if we're not pushing the gas on them, they going faster or slower? Yeah, they're going slower, right? So we have underactive osteoclasts. And remember, means if they're not doing their job, we have low blood calcium because we use them to increase blood calcium, and we have high bone calcium, which is the same thing as if I had too much calcitonin. Kaylin's saying her mind is blown. This analogy, I love this. You all just came up with this in class. I love this. I'm now going to use this forever. So you all are saving students much pain and agony in the future. Props to you all. Parathyroid hormone, that's my gas pedal. It makes those osteoclasts go faster. Calcitonin is my brakes. It slows those osteoclasts down. When you're looking at questions that ask you about too much or too little, think about these things like a gas pedal and the brakes. If I put the gas on, we're going faster. If I slam on the brakes, we're going slower. Normal circumstances, when you're driving like you should be driving, right? Not when we're go-karting and going nuts. Normally, we're either pushing the gas pedal or the brakes to go the speed limit. But if we get all out of control and we're flooring it, or somebody takes out our brakes, we're going really fast. Or if we lose that gas pedal and our brakes are a little too touchy, we're going to go slower. I know it's a lot to process with these too much or too little predictions. How do we feel about them right now? Thumbs up, thumbs down, crying emoji. Barbara says clear as mud. Yep. I would strongly recommend with these questions, I would strongly recommend that you write them down on a blank piece of paper and try to work your way through them again. If we can, because here's what we needed to know to answer these questions. First thing we needed to know is we need to know that we have calcium in the body that's found in two places. It's either in your bones or it's in your blood. When we're talking about the homeostasis for calcium in your body, we're tracking, is it in the bones or is it in the blood? So that's the first thing we need to know. It's either in one place or the other. The second thing we needed to know is whether these hormones were the gas pedal or whether they were the brakes. So if we know that parathyroid hormone is my gas pedal for osteoclasts, that tells me that when it's doing its normal job, I use it to break down bones and get calcium into the bloodstream. And when calcitonin is doing its normal job, it slows those osteoclasts down. It makes me take that calcium that's in the bloodstream and put it into bones because I'm not adding any new stuff to the bloodstream. If you know those basic things about how calcium homeostasis normally works, then just think about driving analogies. So think about flooring it, or think about not having any gas left when you're driving. Think about trying to stop with your brakes, and maybe those brakes aren't working. Use that analogy to help you make those predictions. If I don't have enough, or if I have too much. Yeah, Anisia's got thumbs up and thumbs down, right? We're somewhere in the middle. That's okay. Yeah, let's pump brakes, right? Think it through slowly. Remember, I've mentioned this before. This would be something that I bet when you take your exam, you might want to write yourself some notes about. Remember, at the beginning of your exam, when it first pops up the question, show me your camera that you've got a blank front side of the paper and a blank back side of the paper. If you want to make yourself some notes about this right at the beginning of the test, as soon as you show me that blank paper, I'm okay with that. That might help you to keep things straight. So feel free to use your paper. Once you've shown me it's blank, dump all of this out of your mind onto the paper, right? All right, Kaylin's got a question. Let me read it. Yes, exactly, Kaylin. So calcium in the body is always cycling between two places. Either it's in the bone or it's in the blood. We kind of think of it as we have a finite amount of calcium, and there's two places you find it, either in your bones or in your bloodstream. And it's always going back and forth between the two. Absolutely. It's kind of like how we did the body temperature stuff, right? How we always want to get back to normal. And we can either lose some heat or we can try to conserve our heat. So it's the same thing with calcium. Either when we talk about calcium homeostasis, we're mainly talking about what's going on in your bloodstream. So either I'm going to gain some calcium in my bloodstream by taking it out of the bones, or I'm going to lose some calcium from the bloodstream by putting it into the bones. Yeah, like Kira mentioned, it's totally a negative feedback loop, 100%. Absolutely. So another study pro tip for you, maybe try on a blank piece of paper, try to draw the two homeostasis loops that we talked about. We talked about temperature, and we talked about this calcium homeostasis stuff. So we've got the seesaws, right? They look exactly the same. It's the same format. See if you can fill in those two homeostasis loops when you put them side by side. Maybe seeing them together will help you remember a little bit as well. Yeah, Kaylin mentioned negative feedback loops always working. Absolutely, correct. So we're always trying to keep calcium in the blood at the same level. We will build bone or we'll break down bone to make sure that we stay there where we want to be. Absolutely. So we talked a little bit about osteogenesis imperfecta. Let me get a whiteboard here really fast. Let's briefly talk about our three bone disorders again, though, to make sure we're good on that. We've already talked about osteogenesis imperfecta. We also need to know about osteoporosis. We also need to know about rickets. Okay. So when we talk about these disorders, remember there's kind of, I think I told you this. I know I at least emailed it to a couple of people, but there's a few things that we definitely need to make sure we know about each of these disorders. First one is whether it's calcium or collagen that is my problem. That's the first thing we want to make sure we know. Scoot everything over. Second thing we want to make sure we know is this kids or adults, typically where we diagnose it, in kids or in adults. And one of the last big things that we need to know is this something that's curable? So if you have it, can we fix it, basically? We talked a little bit about this in our other review session, but we've slept at least a little bit, right? Because it was spring break. So let's fill in some stuff here on our table together to make sure we're all on the same page. When we start with osteogenesis imperfecta, we actually already talked about this one, right? Is osteogenesis imperfecta an issue with calcium or collagen? What's my issue in OI? Yeah, exactly. So osteogenesis imperfecta, that's a collagen problem, right? Remember that there is a DNA mutation, so we build bad proteins. There are multiple kinds of osteogenesis imperfecta. We're just learning them kind of as a group. Yes, like Kira mentioned in the chat, remember the parents of the little girl with osteogenesis imperfecta from the video. Don't let them hear you say that it's a calcium problem, right? Because we chatted about this, how people always tell them, well, have you given your daughter milk? Milk doesn't help us with a collagen problem, right? Milk helps us with a calcium problem. Osteogenesis imperfecta is not a calcium problem. So milk, not helpful. Osteogenesis imperfecta, do we typically diagnose that in kids or adults? Kids or adults for OI? Yeah, so we mainly diagnose it, you probably know if you have it in your childhood because your bones break really easily. It can last, it absolutely lasts into adulthood because this one is not curable. Not yet, they're working on it. I think I spelled that wrong. So they're trying to find cures for osteogenesis imperfecta. We don't have a cure yet. What makes it so challenging to cure is the fact that this is an issue with collagen. So this is an issue with a protein that your body makes. I was talking with a student at one point, and they said that they knew someone who had OI, who at one point actually broke their ankle bones just stepping down off of a curb. So it doesn't take much, and you saw that when they were talking about the little girl in the video, right? They were changing her diaper, and she had the baby startle reflex and broke bones. So collagen is really important to making sure your bones don't break. In osteogenesis imperfecta, we don't make good collagen. It's not strong enough, and those bones break. Oh yeah, so Barbara says she's treating a patient that right now, or working with a patient that has osteogenesis imperfecta. Yeah, so it gives you problems with your bones, but it also gives you problems with anywhere else there can be collagen as well. So collagen proteins made poorly. Osteoporosis, this would be another disease. I know Kira had mentioned something about overlapping symptoms. This is a disease that does kind of seem to have some overlap with osteogenesis imperfecta, in that in both of these diseases, bones break really easily, right? So I'm going to put that here in between. Bones break really easily, both in osteogenesis imperfecta and in osteoporosis. Osteoporosis, is that a calcium or collagen problem? Osteoporosis. Yeah, so this is a calcium problem. Absolutely, this is a calcium problem. Osteoporosis in general, do we diagnose that in kids or adults in general? Yeah, for the most part, unless you have some really, really bad genetics, we're going to diagnose this in adults. And again, typically it's going to be postmenopausal for women and later in life for men as well. This is an issue where we don't have enough calcium in the bloodstream, where we start to go after that calcium in our bones. So osteoporosis literally means bones that have pores or that have holes. Osteoporosis. When we talk about osteoporosis, I don't know that we necessarily say it's curable, but there are functional treatments that can help with it. So I'm going to maybe change this to, yeah, like Kira said, I'm going to put here with a star that it's treatable. That there are things we can do to try to reverse it a little bit. Yeah, treatable or manageable. I like that word too. Let me add that word, manageable. What are some of the things that you learned about when you were studying osteoporosis? What are some of the ways that we can kind of manage osteoporosis? Do you remember what some of those things were? Yeah, so we can definitely use calcium supplements, but here's the caveat with calcium supplements, right, is they need to have vitamin D, and that's going to be important when we go next door to Rickets too, right? Here, let me scoot that one over, because we knit for both. I love that several of us mentioned that they actually do use calcitonin supplements. So that hormone that we just talked about, remember that calcitonin, that was my brakes, right? So if we tell our osteoclast cells to slow down, that will help us to make it look like we're building bone faster. So stop breaking down the bone that we have to keep it safe. Yeah, another thing that they recommend, stuff like exercise, because remember that exercise puts strain on your bones, so those osteoblasts get more active. And then there's some of those lifestyle factors, right? So things like no smoking, healthy diet, etc. Those kinds of things help with it as well. So unlike when we talk about osteogenesis imperfecta, where we really don't have a treatment to specifically address the problem, in osteoporosis, we do have ways to try to address the calcium problem, whether it's using calcium supplements, whether it's trying to slow down my bone breakers, or doing some stuff that might encourage my bones to stay strong longer. Last disorder that we read about was rickets. Is rickets a collagen or calcium problem? What's the deal with rickets? Yeah, rickets is also a calcium problem. This is, oops, here, let's try that again. Also a calcium problem. When do we see patients with rickets? Is this typically kids or adults with rickets? Yeah, so this is typically going to be kids. Why is rickets seen in kids and not really in adults? What's special about kids that makes us see rickets in them? Did anyone catch it? Yeah, exactly. So kids are still growing. So they are building lots of bone. If we're building a lot of bone, we need a lot of calcium in the bloodstream for us to be able to do that. So what rickets is basically we don't have enough calcium in the bloodstream. So when we try to build bone, we can't build bone that's sturdy and stable. So when we talk about patients that have rickets, remember you saw in that video, they kind of have bowed legs, or like their skull bends in a little bit. It's because we don't have enough calcium to make those bones really strong, to be able to support a person standing on them. When we talk about rickets, curable or not curable? Can we cure rickets? Do you remember? Yes, we can cure rickets. So this is my one that we can actually cure. Remember, the way that we cure it is with calcium, but especially with vitamin D. That was the key in that video from Canada, right? How these were populations that live far away from the equator, so they're not getting as much vitamin D. We make vitamin D when UV rays hit our skin, and we transform inactive vitamin D into active vitamin D. We need vitamin D to be able to absorb any of the calcium that's in our diet. So this is why when a baby starts drinking cow's milk, they recommend that the baby starts with vitamin D milk, so that all the calcium that's in that milk is actually absorbed. We can't absorb dietary calcium without vitamin D. So we can treat rickets with vitamin D, whether that's in milk, whether that's, like Kaylin mentioned, 15 minutes of sunshine. Making sure we have enough vitamin D will allow us to absorb enough calcium to make our bones strong. So rickets, absolutely treatable, as long as we increase vitamin D. Yeah, and Elapina mentioned a good point as well, too, that breastfed babies often need a vitamin D supplement as well, because there's not a lot of vitamin D in breast milk. So whether it's a supplement or whether it's time outside, that's true for exclusively breastfed babies as well. Absolutely. Any other questions about these three disorders now that we kind of compared them side by side? Yeah, go ahead, Kira. So the question that Kira asked is whether or not OI causes restrictions on activities. I believe that the question is asking, oh goodness, now I'm not 100% remembering that question. Yeah, I would tend to agree with what Elapina is saying. I believe it's asking more about physical activity. So less on the, is it possible for these people to grow up and have a career and have relationships? Absolutely. This is not something that prevents those things, but physical activity, I believe, is what that question is referring to. Yeah, and then it talked about those things. Yeah, so I believe what the question was getting at was the fact that can you do all the same physical activities like someone without osteogenesis imperfecta? And that is a resounding no, right? Fractures way too common, way too easy. That makes it hard to have the same level of physical activity. Yeah, like Elapina mentioned, That girl had so many fractures. She had fractures in utero, right? Remember that? That was crazy. Yeah, another question. Bone question. Kira's question is about intramembranous ossification. Yeah, all we need to know about intramembranous ossification is that it's flat bones that are made that way. Wink, wink, nudge, nudge, that you might want to review from lab what flat bones are. Some examples of flat bones, just so that we're familiar, that flat bones and particular examples of flat bones are made that way. Kira, besides the fact that it's flat bones, you don't have to know anything about it. Yeah, osteoblasts, they do build something called osteoid, which is really weak precursor bone, and then we remodel it into actual bone tissue. But I'm not going to hold you responsible for intramembranous ossification. I would consider the ilium bone a flat bone. Yeah, ilium bone being a flat bone. Yeah, so Elapina's mentioning a question about fingerprints or friction ridges. We talked about those a while back, right? It's been a while. If you really want to completely destroy your fingerprints, you have to go down to that 3rd degree burn. Because remember that those friction ridges come from the interaction of the dermis with the epidermis. Unless I completely rewrite the way that your dermis and epidermis interact with each other, I'm not going to change my fingerprints. So we'd have to do a 3rd degree burn to do that. Yeah. Kaylin asked, on a scale of 1 to 10, how difficult this exam is. Hard for me to say. Now, here's what I think will work to your advantage. Now that you've taken an exam with me, you have an idea of what my exams look like. So I think that is going to help. We obviously have a lot of content. We have a lot of new stuff on this exam. So really maybe a better way to think about the exam is, how does this content compare to the content on the last one? However you feel about the content, the kinds of questions I ask, the way that I ask them is probably about the same as it was on the first one. So if you know your content, if you take your time and read slowly, I think you'll be in good shape. Yeah, so Barbara, we talked about the epiphyseal plate and the epiphyseal line back with bones, but let's review briefly. The epiphyseal plate and the epiphyseal line are made out of different kinds of tissue. What kind of tissue do I find in the epiphyseal plate? Does the epiphyseal plate have cartilage or bone? Yeah, so the epiphyseal plate has cartilage. The epiphyseal line has bone. So the epiphyseal plate, like Kaylin mentioned, means that a person is still growing. If you have an epiphyseal plate, you can see this on an x-ray. You can see that floating space, right, where there's still cartilage and not bone tissue. When a person finishes growing, so 18 to 21, when they finish growing, that turns into bone tissue. And once it turns into bone tissue, we're not growing anymore. We're older. When you look at an x-ray, the bones look normal. There's no floating bones because that cartilage tissue is gone. So epiphyseal plate versus epiphyseal line, it's cartilage tissue versus bone tissue. Yeah, several of us are mentioning that we stopped growing long before 18 to 21. Yeah, so there you go. That 5th grade growth spurt, right? And that was the end. I'm sorry. I think I'd mentioned this to you all. For some reason, I just really wanted to hit 6 feet. I didn't hit 6 feet. that was my one of my life goals, and it didn't happen. So the epiphyseal plate has a place called the ossification zone, where there is some bone tissue in it, but it doesn't completely ossify until it's a line. Most of the epiphyseal plate, if I ask you about the tissue, the epiphyseal plate is cartilage tissue, is how we're going to consider it. Elpina is asking about second-degree burns. For second-degree burns, it's not that the dermis is gone. Third-degree burns, the dermis is gone. Maybe look back at that lab model that showed those layers of burns. In a second-degree burn, we do have damage in the papillary layer of the dermis, possibly down into the reticular layer of the dermis, but we don't destroy it. We just damage it. So the whole dermis wouldn't be gone until we're in the third-degree burn. Otherwise, we would primarily with a second-degree burn, we're going to say that the papillary layer of the dermis and maybe a little bit of that reticular layer of the dermis would be damaged. No, I'm sorry to say, Caitlin, we can't do 20 extra assignments instead of the exam, alas. But you can take that review assignment 20 extra times. The more times you make yourself do that, and the more you make it like the exam environment, the better it's going to be for you. So take advantage. We've said this before. Take advantage of the fact that I use your highest scoring attempt on that assignment. If you feel really good about everything except for the disorders, go through and only answer the disorder questions and see how much you know. Or if you feel really good about everything except the levels of protein structure, just answer the protein structure questions and see what your score is. So go through and get your 90, your 100, and then go through and just do it until you know what you don't know yet. So use that as a way to quiz yourself. So briefly, Temiteo reminded us that we said we talked about CJD. Let's briefly, I'll make us a table here. So we've got sporadic CJD, we've got hereditary CJD, and I'm missing one. What's my last one? I'm forgetting now. Sporadic, hereditary, acquired. Thank you. Yeah, I see your hand up, Temiteo. Did you want to just say your question?
So my question is, for the acquired CJD, there was a question that was asked, how did the anomic prion protein become folded? And for hereditary, the same question, and for sporadic.
Yeah, so it comes down to, for each of these, the big difference we want to know is what causes you to have them in your body. So when we talk about hereditary Creutzfeld-Jakob disease, the emphasis here is on the hereditary part, right? If someone's got hereditary Creutzfeld-Jakob disease, what caused that? Yeah, there we go. So Elvine is mentioning this is caused by a mutation in their genes. And if you remember that video we watched, this was the kind of CJD that Michelle had, right? And so her sons had a 50% chance of developing CJD too. So hereditary Creutzfeld-Jakob disease, there's an issue with the sequence for this prion protein in the DNA, which causes you to build proteins that are wrong. When we talk about acquired CJD, how do we get those proteins that are folded wrong? What's the way we get those? Do we remember? Let me add CJD after it, acquired. Yeah, so Kira mentioned that the main way we really get it is through consuming them. Big picture, if you want to talk about acquired CJD, it just means that the prion came from the environment. The original misfolded protein came from outside the body. That's what acquired means. We acquired it from outside of the body. In hereditary CJD, your body made the protein wrong. So here, I'll add a note here. Body made protein wrong. The original prion came from you in hereditary Creutzfeld-Jakob disease because your genes code for a bad protein. In acquired Creutzfeld-Jakob disease, you bumped into that protein, you consumed it, it came in a graft, it came in through a brain procedure. It was introduced to your body from somewhere outside of your body. When we talk about sporadic Creutzfeld-Jakob disease, what was the deal with this one? Do we remember? This one was a little bit weird. Yeah, so a possibility that Kira mentioned, it's a manufacturing error, or we really don't know. So I put source unknown here. I would put that emoji with the hands up, right, saying we don't know. So in sporadic Creutzfeld-Jakob disease, it could be that you randomly folded protein wrong. It could be that you bumped into it in the environment, but we don't know. We have no idea where. The short answer to sporadic is we don't know why you have it. So either you randomly folded something wrong, maybe you were exposed to it, but we can't trace it. With acquired Creutzfeld-Jakob disease, we can say, oh yeah, you were definitely exposed to it when you had that medical procedure or when you ate that contaminated meat. With sporadic, which is what most of the cases are, we don't know where it came from. We know it's not in your DNA. We don't know a specific place that you bumped into it. We just don't know. That's sporadic Creutzfeld-Jakob disease. So here's your question about manifestations. They all kind of manifest the same way. The big thing to know with Creutzfeld-Jakob disease is that this is a problem with neurons. So those manifestations that we see of it are going to be issues because neurons are being affected. And if neurons are being affected, that's going to affect your motor abilities as well, because the nervous system controls the muscular system. That's what we get to start talking about on Friday. Yeah, so Kira's mentioning that there were a lot of different true-false things that were on the original assignment but not on the review assignment. You don't need to know them in depth for the exam, all the specific manifestations. That's not going to be a big emphasis on the exam. Yeah, that's correct. All right, well, we are over time. I apologize. Thank you all for sticking with me till past the hour. I will mention that I am going to have open office hours tomorrow at 1 o'clock. So if you have any last minute questions, feel free to stop by then. It would also be a good time to consider maybe trying to take the exam in case you have issues, because you'll know I'll be right by the computer then. But remember, the first people to contact if you have issues with the exam, please reach out to Proctorio first, because I cannot get you back into your exam. So if you get kicked out, they're the only ones who can get you back in. Reach out to them right away. If you have a Mac, make sure that you are in, do not disturb, and make sure you don't try to resize your screen. Those are the kind of things that will get you kicked out of the exam. So make sure your window is as big as possible before you begin. Make sure you're not going to get any notifications on your screen from emails or texts or that kind of stuff. You've got 75 minutes, so read your questions slowly. Take your time with it. You should have plenty of time for the exam, and once you finish the exam, then the unit number 3 stuff will pop up for you. So you don't see it now, but as soon as you finish the exam, that will pop up for you. Kaylin asked, is there anything else that I can tell you to help you before tomorrow? My biggest thing is just to take your time. So go slowly with the questions. I do really think that your review assignment is going to be one of the best ways for you to prepare for the exam. So take it as seriously as you can, just like you're taking the exam. Put your notes away. Don't use any assists on that. And then when you finish it, review that attempt and see what topics you need to review a little bit more. So we're kind of into the end game portion of preparing for the exams, right? Because we got to take it by tomorrow. So now is not the time to waste any time in your preparations. So don't build yourself beautiful flashcards. We're probably beyond that. Use that review assignment. Use your notes to go through and focus on the areas that you're a little bit weak. So Caitlin, there won't be muscles on the exam tomorrow. It's just going to be lecture stuff. You're totally fine to write out your DNA and RNA stuff. If you show me your paper at the beginning, I will know that that's what you're working on. And even if it flags you, I go through and I look at recordings that it flags. More often than not, it's just the computer being silly. It's not going to kick you out of the exam for those kind of things. The only thing it's going to kick you out of the exam for is if you try to open another window, if you get a pop-up on your screen, if you leave your exam, that probably wouldn't be great. So those are the kind of things that we kick you out for. But talking to yourself is totally fine. Writing down notes is totally fine. Just remember at the end of your exam, before you click submit, please try to remind yourself to rip up that paper for me to show me that you've ripped it all up. All right, well, I think I'm going to shut off the recording. I'll stick around for just a couple more minutes if we have any last minute questions. Other than that, good luck if you're taking that exam today or tomorrow. You're welcome to email me with any other questions that we didn't get to today. I will do my best to get those replied to as soon as possible, but just take your time on the exam. You got this. We prepared really well for this. I think you're going to do well. So take your time. Don't forget to breathe. You got this.